Okay. Hello, everyone. Thank you for coming to the first seminar of After Matter CMSA seminar of the year. We're delighted that we're having President Josef Käs kick it off. So he's going to be talking about cell and tissue mechanics essential to metastasis. And before we begin, so for normally these are very like lively and interactive. So hopefully, is it okay to ask questions? Would that be all right? Joe?
Yeah, naturally.
Okay, so like if you have any questions on either in person or virtual, you could either just unmute yourself, ask directly. You could type in the chat. I'll be monitoring it. But great. I guess without further ado, let's get started. So thank you. Looking forward.
Yeah, thank you, Joseph.
I said, thank you for the opportunity. Actually, so thanks for listening to me. I changed the title to a much more dull title, Cells and Tissue Mechanics, because that's what I'm doing the last 20 years. So basically, you might conclude, oh, Joseph, again, soft cells, and so he's telling the story he's telling since 20 years. And no, I'm basically trying to get a little bit to the bigger picture for two reasons. Cell and tissue mechanics have become an essential and a very important part of physics of cancer, of physical oncology, and they're definitely a success story. There are students of mine which are now Max Planck directors, professors in Cambridge. So it has become, at least scientifically, an important part. But on the other hand, clinically, it's not relevant. And let's face it, if you do anything with cancer, you have to ask this question, is it clinically relevant? And so what I try to do today is a little bit to convince how we solve this problem that it will become clinically relevant. And so basically there are two parts, two arguments I give you at the beginning. One is up to now, soft matter physics and mechanics were only possible on living tissue, because only on living tissue, you could basically look at moving cells, at dividing cells, and you could do cell mechanics. On a histological slide, it was a big no-no for physics. And I will convince you that the best study object are histological slides. And that means that you can tap in an unlimited resource of data, because from every cancer patient, there's a histological slide. That means
So
90%.
the hesitation was that these histological slides are frozen in time. So you don't see the dynamic.
It's even worse. They're frozen in time and they are fixed in paraffin, so they no reflect the mechanical properties of tissue. So they were totally dead for us. And I tell you how basically you get information out of it, and an unlimited amount of information, because every patient provides samples. So it applies to 92% of all cancer patients, and it's agnostic to molecular details. So it gives you a totally new perspective. And so that's what I'm pitching for. Secondly, the second question is, physics might be simply, okay, we all had our physical oncology centers, but physics might be just unimportant to cancer. So I will convince you that physics is important to cancer, that it basically has the best way is that it's important that it has a diagnostic role, and not just an incremental role, that it has a complementary role to what exists now. So this is basically a little bit the big picture. In that, you will get lots of new soft metaphysics about jamming, about pneumatics. So the nice thing is, it really requires new physics. And so let me start. Wait. Oh, my picture is frozen. Why is that? Okay. So so basically what I want to cover today is challenges in cancer diagnosis. So why do we need physics? And then I have to prove that physics can do it. Then I will talk specifically about cell unjamming, really as a new aspect of soft matter physics. Then I will talk about the extracellular matrix as a tumor promoter, that many people are doing this, but I will show you that describes this best as a nematic phase. And then I will talk about cancer-associated adipocytes, so fat matters to cancer. And then I will try to wrap it up to argue why is this important to cancer.
So forgive my ignorance, but adipocyte is just a fancy name for a fat cell?
Adipocytes is a sophisticated word for fat cell, exactly.
Thank you.
So, and cancer-associated means that it does crosstalk with cancer. Okay. So, so basically, the first, God, we are in the, this Bill Clinton, we are in the century of molecular biology. And molecular biology is definitely at the heart of cancer. So basically the question is, how much information do we have about cancer? So does it require physics in addition, or actually is it just an add-on which gives us money and some new soft metaphysics, but is it important? That is really now basically at least genes, we have billions of data, but currently still we have an oncologist, so basically an MD is facing the following problem. After surgical removal of the tumor, we have now so many treatment options that actually, and there is no sharp criteria what to do. So typically after you remove a breast cancer, you have between 9 and 12 options how you treat it. That leads to tumor boards, which kind of democratically decide what's done. Since it's not objectively decided, the risk of over and under treatment is very high. And that's exactly what you don't want with a life-threatening disease. So basically, everybody's talking about personalized and precision medicine, but if we want to do this, we need reliable prediction and prognosis. And that's exactly what's missing currently.
So Joseph, in Europe or in Germany, the tumor boards are specific to given hospitals. There'll be 6 doctors sitting on this, and they're all from the same hospital, or are they from all over?
At least in every larger clinic, you have a tumor board.
Okay.
In very small, one oncologist alone or one surgeon alone decides.
Okay. Okay. Thank you.
So it's clear. It's part of the standard clinical protocol in Europe, at least. So other things, the simplest and oldest prognostic index is the Nottingham prognostic index, and that takes size, grading, and nodal status. So if lymph nodes are invaded. So very simple thing. It's actually decently reliable, but it doesn't solve over and under treatment. To give you a feel where are its weaknesses. So basically lymph node status is the indicator if there's metastatic risk. We all know this, we ask immediately, so we're all afraid of that. Now, how reliable is it? 30% of all women which have invaded lymph nodes do not develop distant metastasis. 30% of the women which have no lymph node invasion basically develop distant metastasis. So there is a large error of women which can be over and undertreated. So clearly, with 30% wrong predictions, there's a need for new stuff. And naturally, metastasis is a mechanical process, so it will be my primary target. But I will talk about 4 hallmarks of cancer and how it relates to that.
Well, I think there was a question, Joseph.
Can I ask a question? Actually, 2 questions. One is, what is grade of the tumor?
Okay. Grade of the tumor is basically the pathologist looks at it and has several criteria which basically give him a grade which is between 1 and 4.
Okay, so it's just by observation and
it's purely so basically it's not done by a quantitative algorithm. It's done basically by the pathologist who basically looks at the sample, uses his experiences, and tells you how bad or how good it is. In addition to that, you would currently have lymph node status, and the newest thing now is basically gene signatures. Gene signatures are basically the new holy grail of diagnosis. And so we looked at how good are this. For breast cancer, there are things like EndoPredict or OncoPredict, two products. They're very expensive. One test is 2,000 euros. And how well are they doing? And roughly speaking, so we tested 10,000 different gene signatures to really evaluate how good is this working.
So when you say, sorry, Joseph, when you say gene signature, that's some gene expression pattern?
This is basically the expression values of basically of several genes, and we did 10,000 different combinations.
Okay, thank you.
And most of these samples are because they're typically one biopsy. At some instant of time, we don't know how far off they are from when the cancer is metastasized. So whether it invades the lymph nodes or not, or whether it shows up in certain histological sections, is that variability is not accounted for, or it's just there's no way to control for that.
So basically, for all the data I use a data bank provided by the University of Hamburg, which basically is retrospective, so we have the patient histories for 10 years.
Okay, thank you.
So basically, a prospective study always has this problem that you have to wait 10 years. That's why we do it retrospectively, and so I can correlate it directly with patient outcomes, okay?
Thank you.
So the concordance, and here we plotted the concordance. I only plotted a few data, but I could plot here 10,000 different combinations. It drops at 80%. That, roughly speaking, that means 80% of all patients have luck. 20% patients are misdiagnosed. Also interesting. Take more genes. Take longer gene signatures. We could show that 20 genes are optimal. If you take more genes, you're not getting better. So,
sorry, just another naive question for me. When you have to choose the genes, and presumably you could just guess wrong, but you have some sort of intelligent selection of these 20 genes whose expression patterns you're monitoring?
We've done it 3 ways. So at first we used all reported in the literature reported gene signatures.
Okay.
Then secondly, we did something, basically we played lottery. So we took random signatures.
Okay.
And then we used all the current state-of-the-art machine learning algorithms to pick the genes. And consistently in all 3 cohorts, we basically got that maximal performance is between 70 and 80%.
And you're saying the Nottingham, you're comparing now with this Nottingham index with a more sophisticated thing, which you think is more likely to be correct.
The Nottingham index is actually also around this 80%.
I see. Okay.
So basically all these things. So basically we're lacking 20%. That's not so bad, but there is basically there's a whole group of specific patients where the current criteria fail. This is called Greystone patients. So basically there are patients where the hormonal status doesn't help, where exactly the gene signature doesn't help, the grading and the lymph node status doesn't help. So it's not a Greystone where obviously we're lacking information.
And it's not important. We don't have to know what the details of the C index are. It's just some measure
C index. I consciously stayed out of it, but I will immediately explain at least what it means. So,
Yes.
so basically, it's a sophisticated equation, takes variances, blah, blah, blah. And so you can Google it on Wikipedia. But what does it mean? How much information? So how much information is missing if you have a concordance? So we translated this here in basically in a blurred image. So basically here, if you have about 60% noise, that's a concordance of 0.8. So this would be basically a concordance of 0.8. Or in other words, basically in this picture we're currently having, 50% of the information are missing. Now, is this just because we're not looking deep enough, more molecules, or, and that's what I'm basically claiming, that there are emergent effects that cannot be explained as the sum of the molecules. So basically, that there is physics which is not simply explained by the molecular state. And that's what I've tried to convince you in the following. And that's an important thing, because that means physics is not doing this as an inspiration for new soft metaphysics alone. It's also doing this because it will be highly clinically relevant. So the first example is the strongest one, and Jeff really pioneered this. So I'm just somebody who came to this game. Cancer cell unjamming impacts metastatic risk. Here, basically, we have quite a lot of publications. So in that sense, you can read up on that. And so what's the background? Why is it important? So basically, in mature and healthy tissue, that is jammed. So cells can't move because it's jammed. But since it was before in development unjammed, it's close to an unjamming transition. And that means pathological changes, for example, in the lung or in cancer, can cause this unjamming transition. So the unjamming transition is the perfect way to cause cell motility. And so that obviously directly relates to metastasis, and so that's why it's so important, and that's what I want to talk now first. And as I said, it's inherent, so there might be many more diseases where cell unjamming plays a pathological role.
So I think you answered this question maybe last week. Can you remind me, when you have mature healthy tissue, the cells in an epithelial sheet are roughly crystalline? They all have 6 neighbors?
They are not perfect hexagonally packed, but they are very close to hexagonal packing. And you really see the typical hexagonal pattern. So what you see here is, and that's just a piece of, what you see here is on the left is a piece of a tumor. It's a histological slide stained with H&E. And so the darker regions are cells, the brighter regions are extracellular matrix, and it looks very typical. So, and it's inside the tumor, it's not on the boundary of the tumor. Typically, pathologists take always in the inside. And what you see here is, it's full of these cancer cell clusters. So on the right side, you see one of these cancer cell clusters. And naturally, now, if these cells are very closely packed, 90% of the cells are in these clusters. And if you only can escape from the surface, that's a very inefficient process. But only if the cells inside the clusters are motile, you can also have flows to the surface and escapes from the full depth. So basically, then simple scaling argument, if it comes only from the surface, much less cancer cells can escape than it comes from this full volume depth. So basically, cancer cell unjamming, which makes these cells motile, might be an essential process to trigger metastasis.
And I think you told me this already, but I'm going to confirm on the right-hand side, Joseph, we're seeing rather large nuclei inside the boundaries of the cell walls. And that's a feature of healthy cells as well. The size of the nucleus is not a giveaway here.
So, no, it is. So actually, healthy tissue would have much smaller nuclei than cancer cells. So basically, and also what you also see is that there's not a lot of cytoplasm around. So basically, the nuclei will mechanically interact. And that's actually what's currently missing in theory. We have cells interacting, but not nuclei interacting. I will come back to that.
Okay, and then the large nuclei, just as a feature of cancer, whether or not it's metastatic, is due to chromosomal duplication? Or?
It's decondensed nuclei, and they contain simply more DNA.
Okay, thank you.
So already the nucleus, and that is a part of diagnostic. More large nuclei, more DNA, bad prognosis. So now basically I have one jammed, one unjammed. So if you look at it, these cells are sitting close by each other. So they are volume filling. So this is a microscopy slides. So there still might be below the optical resolution some free space. But the free space with these cells, since it's below optical resolution, is less than 5%. So basically they have no space at all. This is above denser sphere packing. And the cell nuclei are a little bit more spaced, as you see. So they are actually below, they behave like colloids, and their packing fraction is below the jamming threshold. Still, that is not fair. That's why you could neglect them, because they're mechanically coupled to each other by the cytoplasm. So they will still strongly interact. So despite being below jamming threshold, they might significantly contribute to the jamming behavior of the cells.
So Joseph, these beautiful pictures where the nuclei are stained red, those are slices through a three-dimensional lump of cells or a quasi-two-dimensional array, or what?
They are basically cut through a three-dimensional vital tumor explant.
Okay. Okay, thank you. And explant just means you've taken it out and you're nurturing it, you're giving it nutrients, and it's in some gel in the lab, or
you stain it with spy DNA and spy actin to visualize them. That's really a revolution now. You can look into live tissue pieces by spy actin and spy DNA staining, and I will show you the motion of that things a little bit later.
Thank you.
So now we have the problem. So basically in 2D, it's hexagonal packing. 3D, it's denser sphere packing. So things cannot move. Here is my simple experiment. So now if I elongate the cells, so basically the hexagons stay solid, but if I have elongated cells, then I fluidize the whole thing. And here, that's naturally a packing effect in my simple simulation. You don't even need a change of packing, because you can do, which you see on the right side, T1 transitions, and the cell can move with basically without changing packing fraction.
So this is
probably somewhere in between.
Sorry, this is a shake table model that you're showing us here? Okay, and then and why do you know why it's always going counterclockwise on the right?
Probably because it was a quick and dirty experiment. It might be slightly tilted.
Okay.
So it was really a quick and dirty experiment.
Yeah, no, fine. It's good. It's very helpful. Thank you.
So I don't conclude too much of it. It's really just to illustrate that shape changes are perfect for unjamming. But shape changes alone, as Lisa Manning would claim, are not sufficient. You also have a nuclear density effect. And I will explain this in a minute. So how does it really look? So on the left side, you see a vital tumor explant. You only see the nuclei, and you see basically in the lower part, in the mid part, you see cells moving. In the upper right part, you see jammed cells. So clearly, in every tumor, in this in the cell clusters, you find unjammed and jammed regions, which you also see again in these nuclei tracks on the right side. So basically, in the lower part, you have an unjammed region. In the upper part, you have a jammed region.
So would you characterize this as a two-phase coexistence, or that's not the right?
I come back to that. Why you have basically why you have jammed and unjammed areas. The jammed areas are rigid cells. The unjammed areas are soft cells. But I will explain this more later.
I can certainly wait.
And these time scales are much shorter than the cell division time. There are no cell divisions going on here, or
good. Thank you, Matt. Thank you for asking this, because naturally also cell division can fluidize and unjam. So basically, this is half an hour. So this is happening on half an hour. The typical scales where cell division would basically unjam that things is more than 30 days. So we are far away from any cell division effect.
So the yellow numbers are minutes then?
Yeah.
Thank you. Minutes and seconds, good.
So you see how dynamic the system really is. And this is probably this is the first slide you ever will see about unjammed cells, and basically previously you tracked single cells in a tumor. We are tracking all the cells. So, and now you see basically here this is a time series of the nuclei getting, and it's squeezing through. And naturally, one thing we're currently doing is we know how stiff the nucleus is, so we can calculate out of the yield stress of the deformed elongated nuclei the local yield stress which is needed that you can start moving. And it clearly shows a yield stress behavior that I can tell you already. And so basically the cells have to be soft enough and strong enough to basically do this. And that means we basically have unjammed cells which move, and we have jammed cells which only perform caged motion. And basically the resting cells have more roundish nuclei, while the moving cells have more elongated nuclei. So simply cells have to squeeze by each other. The unjamming transition helps that all the cells collectively deform each other, elongate, and then can squeeze by each other and can change positions.
So Joseph, just to make sure I understand the middle picture, the green looks like ballistic motion because X is linear in time?
Because it's moving.
And there is some kind of some analog of chemotaxis that's causing it to go in a particular direction.
I probably should. There's also random motion, but this one is in a stream of cells, so they're all flowing in the same direction.
I see. Okay, and then the resting cell doesn't. It flows a little bit, but then it gets jammed, so to speak. That's why
exactly
the blue line rolls over. Thank you.
So now I promised you that you can see all this on a slide which has no mechanical information and no dynamical information. So first thing is what we do is we basically separate the stroma from the cancer cell clusters. In the cancer cell clusters, we determine the cell nucleus shape and the cell shape. And we express this, the nucleus shape as aspect ratio, and out of tradition, we use for the cell shape parameter, which was introduced by Lisa Manning, which is basically the area divided by the volume, or if you go 2D, it's the perimeter divided by the square root of the area. But you could do everything also simply with the aspect ratio. The other thing is dimensionless, but it's the same. And that kind of analysis can be used for 92% of all cancers. All solid tumors are accessible to that. So basically, in this way, we basically can quantify what we're seeing on the slides. And now, basically, if you look at it, so we call this cell and nucleus shape, sinus. And cell and something went wrong here in the formal, so this also should be sinus. So we define sinus as the shape parameter from Lisa, its variance, the aspect ratio of the nucleus, and the variance of it. So basically, we take both things together, because if the nucleus is elongated, also the cell has to be elongated because it's surrounding it. So we basically measure both elongations, and here you clearly see that shape of the nucleus and shape of the cell clearly correlate. They both form the nice straight lines, and so they clearly go together. And so in that sense, they're nicely correlating. And basically, the blue ones are more, in the gemmed region. The green ones are in the blue ones in the ungemmed regions. The green ones are in the gemmed regions. So clearly, we can now use cell shape as an indicator where a cell is moving. If you have an elongated cell shape and an elongated nucleus shape, it's a cell marker for motility.
So I certainly believe this, and it's a beautiful development, but the way you call the green or the blue in the middle graph there is by using explants and literally looking to see whether they're
exactly.
Okay.
So clearly that is done not on slides. That is done on vital samples where you can do cell tracking. But then I say, look, if that correlation is true, I can use shape. Okay.
And is it understood why the boundary between green and blue is tilted away from 45 degrees?
No. So basically, all theory up to now was just concerned with cell shape, and so basically the nuclei effects are really a new player, and so there's lots of room for better theories.
So I have a question about the sinus, the additive sum of the means and the variances of both the cell shape and nucleus shape. So should I understand this as saying that either if the nucleus is elongated or if the cell is elongated, both of them are predictors of motility and unjamming?
Yeah, because first we have gemmed and ungemmed regions, so that gives heterogeneity. Secondly, the cell is deforming, then gets round again. So basically, since it's not always elongated, when it's moving, you get more heterogeneity in the cell nuclei through this succession of round, elongated, round again. That's why we're using that.
Have you all also maybe looked at the ratio of the variance to the mean? So asking how much, exactly as you said. So in the unjammed state, maybe there's more heterogeneity, and so is there other indices that might give you other kinds of information?
Yeah, and actually, yes, there is. Because of lack of time, I'm leaving that part out. Jeff also can say a lot about that. So clearly says there are the scaling laws there. So there's more information there.
Okay, thank you.
I strongly simplify here, just for lack of time.
One more question, please, Joseph, here.
Yeah.
So I think a few slides ago, you mentioned that the cells that are jammed are stiffer effectively than the unjammed cells.
I come back to that. I will show you.
Okay, and you relate that to this shape parameter or shape index? Is that
currently the shape parameter is basically a mixture out of how soft the cell is, how adhesive it is, and how much pulling force it has. So it's a mixture. It's a phenologic. It's basically a very coarse-grained parameter, and I will discuss how the different real effects of a cell play a role a little bit later.
So the Nottingham index, which you showed us at the beginning, had this 0.4 or 0.4 factor, you could do that with this sum as well, right? I mean,
Yeah.
you say very phenomenological.
And I will do this.
Okay, good, good. And then just to get the intuition for the picture on the right, the mean velocity as a function of cell volume. So it's going faster for large cell volumes? Is that?
Yeah, and
How do I
thank you.
understand that?
This is, so basically, so the left side is shape, the right side is density. So while there are no density cell packing effects, the nuclear spacing plays a role. So nuclear number density plays a role. And nuclear number density is directly proportional to cell volume, because the cytoplasm around spaces the nuclei. And so what you see here is that if there's more space between the nuclei, the cells are getting faster.
I see. So that's
we have basically two effects. We have a shape effect and we have a nuclear density effect.
Okay.
I have a quick question. How is the 3D shape index defined and measured?
So,
I'm just going to take 2D.
So basically, the shape index in 3D is basically the area, the surface area divided by the by the volume in some way that it's dimensionless.
Yeah, so 2/3 power of pi.
Yeah, exactly. Thank you. Thank you.
So here, if you wanted to do ellipsoids, you'd have 3 principal axes, and this other alternative is probably a little cleaner, I guess, in 3D.
Yeah, and we're now, the data we are using are 2D data because we're going to this a lot. So we are basically not as accurate. So we say that 2D data reflect 3D data. There's a nice paper from Andrea Liu that this matches, but you make errors and we accept these errors.
Okay, okay, thank you.
So now, many phase diagrams of cancer cell unjamming are in the literature. And they all got it wrong. They got it wrong because there was a ridiculous fight if it's shape induced or density induced. So what I'm suggesting, it's both. So one axis is the cell area, which is basically nuclear density. And the other one is not the shape index, it's the CNOS, the cell and nucleus, the cell and nucleus shape, the cell and nucleus shape index. Something went wrong on the slides. And so in this way, we basically took from 1,400 patients the histological slides and placed them in this diagram. And clearly, so basically this is reflecting exactly the spectrum of shapes. And if you look at the right side, basically it shows all the 1,380 samples and how they distribute. To the boundary and the triangles and the blue dots, I'm coming back later. But what I'm saying is that if you want to have the whole, I'm not saying phase diagram because it's a metastable state, so I'm saying state diagram.
Yes.
So if you want to have the real state diagram, you basically have besides one more is yield stress, but so there's a third axis. But in the static picture, you have basically 2 factors: the packing of the nuclei, which we express by the cell area, and this elongated cell and nucleus shape, which we express by CNOS, by the shape index. And so you have 2 axes. And now you have this beauty with 1,380 patients, you really catch the whole phase space.
So just make sure I understand, the 1,380 patients, for each patient, you take these sections from the tumor, and then you average over all the cells that are in that section for a particular patient, and then you put that on this diagram.
No, we actually don't average. We basically take we take the cancer cell clusters and classifies them and then puts them into this thing. So it's even more detailed.
Okay, and then these patients presumably come from a variety of ages. So could it be that there's a different state diagram for women in their 60s as opposed to women in their 40s? Has that been looked at?
So first question is, God, if everybody might have received already some treatment and so on. So number one is we basically had one cohort of patients, 400 patients, which only received surgical resection of the tumor. And the problem is that they are the ones with good prognosis. Then the rest of the patients was basically all kind of chemotherapy and whatever.
Yes.
And so basically not all of the patients might have developed distant metastasis, despite that we really use distant metastasis to verify that we catch the motile cells on the right side, because it requires motility.
Yes.
So basically this is an error. So we might underestimate the motile cells, but still most of the things which metastasize we got right. So in that sense, there's a clear correlation, and I will come back to that.
Thank you.
So, and there is a problem before somebody realizes it. I didn't put in a transition needs a boundary. I naturally don't have a sharp boundary because theory doesn't apply. And so we're currently using machine learning and this explants to find a sharp boundary. And roughly speaking, the dotted lines represent this kind of boundary, but that's still phenological. So we're still looking for the boundary, and if it's first order and so on. So there's still physics, the transition physics is a little bit missing in our diagram, but at least we are proposing the right variables for state diagram.
And then just make sure I'm still on your wavelength here. So the picture on the right and the picture on the left have the same axes, but what's different about it? That the raw data is more on the right, and then you've dressed it up somehow on the left, or what's going on?
No, basically we translated each data point here in a drawing how a cell would look like.
I see.
The left side shows you how the cell looks like in this thing, and the right side shows you if you find a tumor sample in this area.
But presumably there are many shapes of cells that are all roughly the same that would be consistent with a given data point on the right.
Yeah, which you clearly see. It really clusters here.
Okay, all right. Thank you.
So basically the idea was also to catch the exotic stuff. Okay, so basically clearly I hope that I convinced you that there are two drivers of cell jamming and unjamming. One is cell and nucleus shape, and the other one is nuclear density. But why? How does it come from? And now, first thing is, you already asked sometimes, what role does cell elasticity play? So what we did is we used an AFM on tumor explants. We simultaneously stained the nuclei that we could see where the nuclei are. So basically, we made sure that we only do AFMs on cell clusters and not on extracellular matrix. So basically, we always looked at the areas where there's jamming and unjamming, and not at other irrelevant areas. And so the picture you will see here, that's breast cancer. On the left side, you see healthy, and that's all just very soft because there are not very much epithelial cells in a normal breast. It's all fat cells and they're soft. Now we come to the tumor cluster, and the tumor cluster, we have these rigid islands in a sea of soft cells. These are all cancer cells. So clearly what we see here is that basically the tumor is heterogeneous. It has a significant fraction of rigid cells in red and soft cells in blue, and the blue cells prevail. So we have more ungemmed cells than gemmed cells. So we're completely, so basically we have this heterogeneous state all over. If we average, surprisingly, this still gives you a mechanic. So also, if you measure, if you indent the whole spheroid, then you get a finite G prime. So these things are still mechanically stable. Despite that we only have solid islands in a fluid, we get a finite G prime and a mechanically stable behavior. And if you average over everything, the tumor is stiffer than the fat cells.
And can you measure the G double prime as well?
Yeah, but we haven't done anything with that. There is, roughly speaking, actually G double prime might be even more important because you basically have a solid with lots of dissipation.
Yep. That's what I was wondering. And I think, again, Max may have asked you this already last week, but the middle, the second picture from the left, are the red stiffer cells percolating in 3D? Do you know?
I don't know it, but from the fraction, so basically from the fraction of cells we count, they are below the percolation threshold. I will come back to that in a minute.
Okay, thank you.
So, but if you look now, if you compare here tumor cells to their predecessor cells, to the epithelial cells, not to the fat cells, then clearly they form this soft tail. So there is basically a highly heterogeneous distribution with rigid cells, but also with soft cells. So one unjamming reason might be very well that the soft cells are unjammed. How
So
do we?
Okay. Oh, sorry.
Yeah.
Can you remind me what cell type is FA and HME?
FA and HMM is basically epithelial cells from a benign tumor or from breast reductions.
Okay, I see. But that will be healthy. The breast
That will be
reduction
healthy.
Healthy. Okay, thank you.
I just separated. So the larger one is breast reduction. The other one is fibroadenoma, which is a lump, but a harmless one.
Thank you.
So same with cervix cancer. But cervix cancer, the main difference is the cervix tissue, the control tissue, is much stiffer because it's full of epithelial cells. And again, we have the island of rigid cells in a sea of soft cells. But that all tumors are stiffer than their surrounding, we disproves this here. Here, the healthy tissue is stiffer than the tumor tissue.
And it's a separate talk, so you're probably not going to get into it, but you're actually able to measure these moduli locally, cell by cell, or clusters of cell by cell?
Yeah. We know where the nuclei are, and then
so you just do it from the nuclei.
Yeah.
Okay, thank you.
And this is like with an AFM style measurement, or is it?
So we did both. We did the measurements with AFM because that gives us spatial correlation, and we did optical structure measurements in addition.
I see. Okay.
And when you do
and we did magnetic resonance elastography to get it on the large scale. So we basically had three ways to measure it, and all three ways are consistent.
And do you ever have a use for confocal microscopy as well in these?
Yeah, basically we saw the nuclei. So we have this fancy AFM where we can see nuclei with fluorescence, and then we could even align them afterwards because we know where the nuclei were with the confocal images in the fixed samples. You see cell softening, and so we did vertex. So basically, Max B did vertex simulations, and it's very interesting. So basically, if you have more than 60% rigid cells, you get a percolated backbone, and this is giving a solid behavior. Then if you have if you have basically if you have much less, then if you have basically only a very small fraction of rigid cells, you get fluid behavior. But the tumors, so basically the solid behavior is exactly the behavior we found for the healthy tissue. But the tumors are exactly in this regime we call heterogeneous solid regime. And there we only have a fraction of about 25% rigid cells. So basically only 25% of the cells form jammed islands, which clearly cannot be percolated. So we really have islands of jammed cells in a fluid sea of soft cells.
So
Now, how does it still give a finite G prime and a mechanically stable state? That's happening due to tension percolation. So in the simulations, the red stuff is how tension percolates in the systems. And so,
okay, good. And Max P is doing this in 2D
in 2D and 3D.
and 3D.
So basically, this is shown in 2D because it's easier to show. But we basically also has more elaborate simulations in 3D.
Okay, and then the heterogeneous solid, are we tempted to think of that as being like a two-phase region between solid and fluid?
I would say so, yeah.
You would or you would not?
I would, I agree,
Oh.
I agree.
Okay, thank you.
So I agree, and so basically, so clearly, and the interesting thing is really now the tumor has the best of both worlds. It's still mechanically stable, has a finite G prime, which is actually large enough to push the rest out of the way, and it has all the motile cells which are soft and unjammed. So it's a perfect state for a tumor.
Perfect from the point of view of metastasis, but not
Exactly. Sorry for it's a very disadvantageous state for us, but for cancer, it's a pretty cool thing.
We're very happy.
Okay, can I ask a clarification and question? So, did I understand correctly that you said in the heterogeneous solid, the rigid, the red or the harder rigid cells, they don't percolate, but tension percolates?
Yeah, exactly.
And so you have tension percolation that precedes the actual percolation of the cells themselves.
Yeah.
And that.
So you don't have a solid backbone, but you have a percolated backbone by a tension percolation.
And that is, what's the intuition for that? Because in networks, it's exactly the opposite, right? You first have particle or node percolation and then rigidity percolation. So here you seem to flip it. And is there an intuition for this?
So basically, so first is we find it. So we really find it experimentally. So we find the fluid sea, we find the low fraction, and we still find a finite bulk shear modulus G prime. So basically, that's what we find. And then basically, since they are not percolated as a solid backbone, I think it only can be a tension percolation. That's the only thing. And then we tune the simulations towards that direction. So if anybody has a better idea, I'm happy with that. So I'm just telling you, we have a macroscopic solid behavior in something which persists out of 75% fluid.
That is fantastic. I think it's really nice, and I'm just curious to know if there are if there's an understanding of it. But anyways, yes.
So I'm careful. If I could give you tension percolation is basically a flashy word, and it has to be something like, so there has to be some mechanical coupling. The vertex model is strongly simplified. So in that sense, there might be more to that truth. So basically what I'm confident about and what I can prove is that we have a new state where basically a fluid behaves like a solid.
And for the uninitiated, tension percolation is the same as rigidity percolation?
Yeah.
Is it possible to mimic this in a passive system of grains immersed in a fluid?
We haven't tried that. But I like your idea.
Thank you.
So clearly cool experiment. So, but sorry, there's so many things to do.
Yes.
So in the simulations, you're not playing with cell adhesion or obviously it's 2D, so no cell substrate adhesion. So you're just changing sort of the elastic parameter.
So we basically, what we do is we take the real distributions we measured before, and we basically take the threshold of the median value of the healthy cells as a separator to separate between stiff and soft cells, and then we basically have one soft cell type and one stiff cell type.
And have you looked at density of disclinations as you unjam the system?
No, but I would like to do that.
Okay. Thanks.
So next thing is, so we had stiffness. Cell adhesion is next thing. First is tissue surface tension in the systems nearly doesn't play a role. In healthy tissues, you have a contractile ring around, and that's really pulling everything together, and you have high tissue surface tension. In the cancerous systems, there are not these rings, so there's only cell adhesion, which is lower, but still it plays a role. Basically, there are epithelial cancer cells and mesenchymal cancer cells. In the epithelial, there is ecatorin, and there you basically get collective cancer cell escape. So you see, basically you see these pneumatic phases which are streaming, and here you see it also, and then basically whole fingers are budding off. While if you have lower adhesion, no ecatorin, then basically the cells are moving randomly and single cells escape. The first thing is the more dangerous one.
So the just remind me, please, E-cadherin is a protein that spans two neighboring cell walls.
Exactly. Ecatorin and desmosomes are the two cross-linkers between epithelial cells, typically. And ecatorin is in cancer replaced by N and P caterins, which are weaker.
I see. And that helps, of course, their motility and their ability to metastasize. And are there ways of like hitchhiker mutations that are associated with changing the type of cadherin?
More complicated. The real expert you have right around the corner, Herb Levine. So basically, Herb showed that there is no epithelial mesenchymal transformation. So cancer cells are not transforming from E to non-ecatorin. They basically show a spectrum of mixed states, because that's the most advantage. So you find these more aggressive cells which still have ecatorin, and you find the cells which are basically more fluid with less ecatorin. So you find both, and you find a wild mixture, so a whole spectrum, which naturally makes it now very hard to talk about any characteristic mutations which drives that.
I could just interject.
Thanks.
About a year ago, Andrew Ewald published a paper in Nature that showed that in mice, E-cadherin is necessary for metastasis, which is totally the opposite of what everyone thought. Everyone thought that you have to lose E-cadherin, like in an EMT, so that the cells were free to migrate as individual cells.
Yes.
But he found precisely the opposite, that E-cadherin is necessary for metastasis. So that was in Nature. Ewald, E-W-A-L-D.
Thank you.
Yeah.
So same story here in the Nature Cell Biology. We also say it needs epithelial type cancer cells because they are budding off collectively, and that's more aggressive. Okay, so are we able to do something which is better than current prognosis? So basically we drew a boundary. So below below you find the more densely packed cells and the more roundish cells. And above you find, in our perspective, unjammed cells. And so we correlated this with metastasis, and you clearly see that most of the triangles are above our boundary. So clearly, unjamming correlates with metastatic risk. Okay, that's nice for the physics.
Okay, so now if I just look at the blue dots, that's lymph node positive, the cancer cells in the lymph nodes, and that's on both sides of the boundary, but you're saying it's predominantly above? So
basically, if you see, the lymph node also is diagnostic, but it's much worse. So basically, all the blue lymph nodes which don't have a triangle are false positives.
Yes. Yes.
And all the triangles who don't have a blue dot are false negatives. So you clearly see that sinus outperforms the nodal status. So we're doing better than that.
Oh, I see.
So basically, we have a better prognostic factor than nodal status. So physics is important, unjamming is important, and we show this here in this Kaplan-Meier plot that it clearly separates survival, high risk and low risk group. So this is that jamming part. Now extracellular matrix, so much more people are talking about, so I will keep it short. So basically, extracellular matrix is known. Inflammation causes fibrosis. So ECM is drastically enhanced. That means the ECM is stiffer, which acts through mechanotransduction as a tumor promoter.
So what does fibrosis mean? Just a proliferation of fibers?
Fibrosis means more collagen fibers. Since basically the fibroblasts, which are around cancer, are stimulated to produce more collagen.
I see.
And that stiffer matrix, then basically Paul and Valerie Weaver pioneered that. Basically, if a cancer cell sees a stiff substrate, it becomes really nasty. So, and what I want to suggest is, so basically, how can you quantify this in this histological slides? So, very simply, normal ECM is isotropic. It's a polymer network out of collagen fibers. Now, if you condense it, since they have a high persistence length, they perform a nematic transition and get aligned. And we can measure these fibers and we can measure their alignment, and that's what you're seeing here. So they clearly form a nematic, and we can follow the nematic director very well. So what I'm suggesting is to describe fibrosis, to describe it as a nematic phase, and so to describe so basically how is it oriented towards the cells, how dense is it, how many defects are there, clearly should be an indicator. And I will show you immediately that describing this as nematic gives you a prognostic factor.
But since this phase, this extracellular matrix comes from fibroblasts, you're sort of trying to read the mind of the fibroblasts, so to speak. They're the ones that are somehow.
They're much less fibroblasts. So they're basically probably 1% of the space is filled by fibroblasts.
Okay. But the argument here is there's a phase transition or a state transition of some kind in the extracellular matrix that is also a diagnostic for cancer.
Exactly. So if you have fibrosis, you will find a nematic. If you have healthy ECM, it will be more isotropic.
Okay. Thank you.
And maybe I missed this, but does fibrosis precede metastasis, or was it after? Is there a
fibrosis promotes all 11 hallmarks of cancer. So it is clearly a metastasis promoter, and honestly, I've never seen so early tumors that there wasn't fibrosis. So basically, in all my slides, I haven't seen tumors which didn't have unjammed cells or fibrosis. So don't ask me what comes first.
Okay. And is there an interaction with the immune system in terms of as with the buildup of ECM to neutrophils or other immune cells?
So basically, jamming. So basically, jamming and unjamming and fibrosis happens earlier than basically the switching of the immune system. That's happening right now.
Okay. Thank you.
And the scale bars on these pictures, the HD picture, for example, what are the black bars again?
What?
What are the scale bars?
The scale bars here are about 100 microns. Sorry, I should have.
Okay.
So what you see is clearly this is an area where we have a low density of collagen, so isotropic, and you see how the tumor spreads as single cells. And in the high density of somatic, which clearly cages this thing, you see this much more collective growth figures. And so, which is more aggressive. So clearly collagen density also basically is the nematic basically determines how the cancer cells can invade it. And naturally, the simplest thing is if the nematic is aligned radially to your cancer cell clusters, that helps. If it's basically parallel to the surface of your cluster, it's like a cage and inhibits. And so that's exactly what we tested first. So we took the nematic order parameter and we basically stratified in two groups. If the order parameter is perpendicular to the cluster boundary, that's high risk because it's like a transport highway to the outside, which also tumor biology predicts. Or if it's low risk, then it's parallel to the cluster boundary. And we classified our 1,400 samples this way, and it clearly stratifies. So really, if basically your nematic is going readily out, you have a bad prognosis. If it's basically like a cage surrounding, then you have a good prognosis.
But presumably,
again, you find a new prognostic parameter by using this nematic transition.
But presumably, the actual boundary condition could be tilted in between parallel and perpendicular.
It's strongly simplified. This was a first try. You're absolutely right. And next slide is exactly saying this. Naturally, I don't consider defects here, and defects could have a tremendous effect where you can grow and where you can't grow. So in that sense, there's much more to the nematic. There's much more to the nematic. The nematic director is just what you get immediately. And so this is a first try and not the end of what you can do with this nematic description.
So Joseph, you've been very patient with our questions. Maybe you could finish up in five minutes or so. Is that...
I can do this. Luckily, I can do this. It's getting shorter and shorter.
Thank you.
So one thing, I basically don't go into the details because it's anywhere more biology, but it's not only the connective tissue, the collagen matrix, which modulates. Basically, every cancer, most cancers are also surrounded or in contact with fatty tissue, and the fatty tissue is also modulating the mechanical properties. How it's doing this is biology. The adipocytes send out, the fat cells send out exosomes. The exosomes cause the formation of lipid droplets in cancer. That changes the lipid composition, and that changes cell mechanics. So this is clearly biology. What comes out of it is interesting because there is fat, which is a tumor inhibitor, so which makes cancer cells stiffer, and there's fat, which makes tumor cells softer and more motile. So that is really a difference. So because you also have tumor downregulators. This is very unique, and that even basically goes further. If you look at different fatty tissue around the cervix tumor, there are repulsive ones and attractive ones. The ones which are early in development are repulsive. The ones which are late in development are attractive. which kind of gives us hope, because this seems to be a lipid story. And there are lots of medical drugs where you can change lipid composition, which are usually used for heart and vasculature. And there might be a totally new way that you basically make tissue repulsive to cancer. And that's a more biological story. That's why I keep it short. Still, it's mechanical changes. As I showed you before, these are mechanical changes. So again, it fits into this raster that it needs physics. And I want to end now that naturally one thing I haven't done, which is naturally dear to David, evolutionary and spatial dynamics, you also can do on the slides. So basically, the tumor biologists are talking about infiltrative growth, expansive growth. The expansive growth is high proliferation. The infiltrative growth is lower proliferation, but highly metastatic. But the expansive growth is considered more aggressive. Now, what we can do is, in the slides, we can quantify proliferation in the slides with proliferation markers, low and high in clustering, and we can basically quantify cell motility. So we basically have all the tools that you can model this growth, as basically Max and David has done already, and which has been done frequently for bacterial system. And again, I'm sure that we will get a much better metastatic predictor than we have now. So in conclusion, we basically have 3 or 4 hallmarks of cancer where physics involved: uncontrolled growth, metastasis, genetic instability, and inflammation. And now, why is it cool to do it with physics? Because we find the quantitative relationships between proliferation, cell motility, fibrosis, and genetic evolution. By finding a so basically, I told you, currently pathologists do this by the eye. If you find a quantifiable matrix to do diagnosis, this alone is already a big advantage. In addition, if you get the context, if you get the relations quantitatively right, then you're not stratifying by life or death, you can stratify by these hallmarks of cancer. And that means you will have a much more stratified prognosis, which also will help you to much better therapy decisions. And that's where I want to end. And I have an incredible group. Paul is not part of the group, but he's a always well-seen visitor, and I have great, and I basically have a lot of collaborators, and so thank you for listening.
Thank you.
Thank you for the wonderful talk. We've had we've had many questions, but if there are there any further questions, either from Zoom audience or in person?
There's been so many that have been asked. I suspect they would be urgent.
Check.
Any Zoom questions? Zoom people can speak up if you have a question. Hey. So this has been wonderful, Joseph. Thank you particularly for your patience with a lot of the questions that have been coming at you. And I'll email you a place where you and Max and I can get together and maybe follow up perhaps in about 10 minutes. Would that work for you, Joseph?
Works for me. I sent you already a link.
Okay.